I'd like to welcome everyone to today's session. It's my great pleasure to welcome this morning the winner of the 2023 Translational Microbiology Prize, Professor Ravindra Gupta of the Cambridge Institute for Therapeutic Immunology and Infectious Disease. The Translational Microbiology Prize is awarded to an individual who has demonstrated an outstanding contribution to translational microbiology. Ravindra more than meets this criteria. His lab has worked extensively in HIV drug resistance, both at the molecular and population levels, and has contributed to the appreciation of the scale of drug resistance globally. Also working on SARS-CoV-2, his team is characterizing the virology of key spike protein mutations in new variants and their impact on natural and vaccine-induced immunity. It is my great pleasure to welcome Ravindra to talk to us about SARS-CoV-2 variants and vaccines.
Good morning, everybody, and thank you so much for joining me this morning for this talk. And I'd like to thank the Microbiology Society for this great honour, and I'm very pleased to accept it on behalf of myself, my current team members, my mentors over the years, and I'm extremely proud to be here to present a talk on SARS-CoV-2 variants and vaccines. So I'd just like to start by thanking some of my mentors over the years, and I know Greg is sitting in the audience here, but I started my career as a medic wanting to do research on public health problems and thought that HIV drug resistance was going to be an issue because of the massive scale-up of ARVs globally for a virus that was more mutagenic than anything we'd appreciated before. And so I spent many years trying to characterize this, and really this work led to changes in WHO guidelines for treating HIV. But at the same time, at UCL, I was having lab meetings with Greg Towers, and I couldn't help to be fascinated by the work on innate immunity and host restriction and how viruses are evolving alongside pathogens to overcome them. And so I ended up doing a sort of joint PhD and really getting stuck into molecular virology as well as all this public health related work. And really that gave me the right ingredients for my future research career. And I think that's a message I'd like to convey to the younger scientists among you, in other words, to pursue the interesting things that you want to address, no matter which skill sets you may need to look for to answer those questions. And finally here, Paul Lehner at Cambridge, who has been a mentor more recently during the SARS-CoV-2 pandemic, and also as I moved from UCL to Cambridge, has really been pivotal in my settling in there. I'd also like to just acknowledge my team members. These are individuals who have been with me for many, many years and were here during the SARS-CoV-2 pandemic. Some of them have now moved on. But of course, without your team, there's very little you can do. And so the work I'm going to show you will be their work, really. And this figure just shows you some of the contributions we made during the pandemic. I would like to say that initially we were very reluctant to enter this field because we're not respiratory virologists. We were working on HIV, a chronic infection, and of course we thought this was going to be a coronavirus-like disease with a short incubation period, a respiratory pathogen, and so we didn't really know what we could do, and actually ended up pivoting our lab to this largely because of clinical questions arising from both public health and the clinic. And again, that fueled our interest and passion and desire to really answer key questions about this virus. And of course, now it's become the most interesting virus that I can think of, and that's why we've moved largely from HIV now to studying SARS, because we can watch its real-time evolution in front of our eyes, whereas with HIV we would have to wait much longer to see how this virus was changing. So in terms of an overview of this talk, I'll give you a bit of background on virus-cell interactions. Much of this you'll know already, but I'm hoping to add a bit of detail. Some insights into evolution from colleagues around the world. I'd like to talk about how new variants are arising, because I think that's a really big issue to address. And then to spend some time on biological characteristics of variants of concern, because again, we're watching real-time evolution and adaptation to a host. and then to look at some of the challenges, both in terms of vaccines and future directions as to how we may address this problem long term. So let's start let's start with understanding how the virus engages with the cell, and Wendy introduced some of this yesterday. Of course, you all know the spike protein is sitting on the surface of this enveloped virus with its RNA genetic material in there. The spike protein engages with ACE2 on the surface cell, and of course ACE2 is very, very widely expressed, and this is one of the key characteristics of SARS-CoV-2, of course, and we believe this is part of the reason why we have chronic infection. So once that engagement has occurred, you get a series of conformational changes that leads to a fusion peptide exposure and fusion of viral membranes. The key thing actually is to appreciate that the spike is a trimer, and this is how it's arranged on the surface of a virion, so it's very irregular. Because it's a trimer, it can be in different conformations, and the receptor binding domain, which engages ACE2, can be found in both an open and a closed conformation, and it can only engage ACE2 when it's in the open conformation. So you can have a situation where one receptor binding domain is up and open and can engage, and if none of them are open, then this spike protein can't engage ACE2. And the reason it's flipping up and down is because, of course, this is an immunodominant epitope and a target for neutralizing antibodies. So the virus does not want to have exposure of these epitopes unless it's absolutely necessary. So the virus is playing a balancing act between viral entry efficiency and escape from neutralization. So this is a closer look at that interface between the receptor binding domain of SARS-CoV-2 and ACE2. And what I've highlighted here are key, some of the early escape mutations for neutralizing antibodies, E484, changes at 417. And you can see that they're sitting at the interface between the virus and cell. Of course, this highlights the fact that the virus is trying to escape from neutralizing antibodies by making these mutations, and whilst it has to maintain electrostatic interactions between itself and ACE2 in order to achieve entry efficiency. So this sets up a very complex evolutionary situation, and the variants of concern have really elucidated what's going on. So before we move on, I just want to show you in a little bit more detail what's happening with the spike protein when it engages ACE2 through this nice figure. So you can see here ACE2 engaging the up conformation of the spike protein. This leads to a rearrangement, so quite a dramatic rearrangement of the spike protein. And we see this with other enveloped glycoproteins such as HIV-1. And that leads to shedding of S1 and exposure of this fusion peptide, which is part of the S2 part of the spike protein, and that allows, and this is a hydrophobic, it inserts into the target cell, and again, further rearrangements draw together these two membranes until you get fusion of these phospholipid bilayers. So it's quite a complex, beautiful piece of machinery that's highly conserved in evolution. And indeed, humans have co-opted this process, for example, to form syncytia and, for example, in placenta. So going back to the evolution of viruses, and in order to understand how we dealt with SARS-CoV-2, let's go back to a classical sort of figure that's in the textbooks, which demonstrates evolution of viruses through two mechanisms or two paradigms, drift and shift. And here you can see this is a segmented virus as an example. This is, for example, influenza. And the drift is accounted for by single mutations that happen over time, mainly due to polymerase errors, and beneficial ones are selected in the host. But of course, you have this alternative, more radical way of generating diversity, and this is shifting. For influenza, this is achieved through a process of exchange of material between two viruses to generate a recombinant virus in the producer cell. So you get a cell infected by two different viruses, virus A, virus B, and then you get a new virus coming out. And this has got a mixture of segments, and that leads to much more diversification, and therefore the ability to escape antibodies that have been evolving against either of the original strains. So that's shifting. And so when SARS came along, we tried to apply our current paradigms. And early on, we estimated a mutation rate of 2 substitutions per month, which is relatively modest. And this fitted with a short incubation period that we were observing, a lot of asymptomatic transmission with high infection turnover, high numbers of cells, and a sort of hit-and-run approach. In other words, infecting an individual, moving on before symptoms have occurred, and coupled with proofreading of the RNA polymerase, this should all have led to relatively low selection pressure on the virus to mutate because it was already so efficient. And of course, shifting was unlikely, we thought, because this virus did not have a segmented genome, unlike influenza, and therefore we didn't think that antigenic shifting was going to happen. And the early evolution did justify this theory, because if you look at the sampling dates and the genetic distance of SARS-CoV-2 early viruses, you could see that they were, in fact, drifting with a consistent mutation rate. And shown here is a next-clade plot. This is a straight line that you'd expect, but of course you could see that there was deviation occurring from that straight line. So the substitution rate had clearly changed with the arrival of variants of concern. So the question here is, what's going on? Well, if you look at the phylogeny, you'll see that these variants of concern arise from these very long stem branches. And we'll discuss where this evolution is happening. But of course, once you've achieved a variant of concern, the evolution rate actually is very similar to the basal lineage back here. So the rate of evolution once you've established a variant of concern is actually quite similar. But the difference is that if you look at the rate of evolution in the stem, here you can see that it's elevated. So really the adaptation is happening here. The acceleration of selection, you may say, is happening along that stem branch. And Richard Neher has shown this very nicely. This plot shows you the interclade synonymous rate, which has been fairly stable and at around 6 substitutions per year. But if you look at the amino acid rate, it's much, much higher. Between clades, and this is telling us that the DN-DS ratio, or the rate of positive selection, is much, much higher than the rate of substitutions that don't lead to coding changes. So what's happening is that there is evidence of positive selection and adaptation between variants of concern, or clades. So the result of all of that is really is antigenic shifting. We didn't think it was going to happen, but in fact it has, of course. And this is a plot showing you how variants are mapping in 2-dimensional space relative to each other, and this is all based on neutralization. So this is taking sera from individuals who have been infected or vaccinated, and looking at how far or how well they're neutralized by sera, and this is applied to different variants of concern. Of course, in 2021 we had Omicron out here, and the original Wuhan virus over here in blue. So this was at that time the greatest known distance. If you fast forward now to 2022, 2023, you can see that Omicron BA1 that I showed you is now in the company of its relatives, and you can see that actually we've got very, very significant diversification and antigenic shifting, even post-Omicron. So the Omicron lineage is not the end of an evolutionary process, it's actually the beginning of a new set of shifting events. So we will discuss why that may be a little bit later on. So SARS-CoV-2 appears to be able to have shifted antigenically, we think, to avoid neutralizing antibodies based on existing data and data from other viruses. But then the question is, how has that happened? I've said that there is no segmented genome. There is proofreading ability of the RNA polymerase. So how is it achieving this? And there are 3 main hypotheses about this. One is that you could have transmission of the virus through lots and lots of people, and the virus is picking up individual mutations in between individuals, and that they're going undetected over long periods of time. Now, of course, with the alpha variant, we had greater than 80% genomic surveillance coverage in this country, and we didn't really see individual intermediates of the alpha variant. We did see an intermediate that had about 4 or 5 of the mutations in alpha in a single individual, but certainly we didn't see any evidence of this happening. And then, of course, the data I just showed you, showing you that the substitution rates for amino acids is much, much greater than for nucleotides. This also goes against this process, because you would expect the mutation rate to be the same for both nucleotides as well as amino acids. The second option is considering spillover into wildlife and then a spill back into humans. We have seen variants of concern circulating in animals, certainly, but I think that in terms of the number of events that would have to happen, this, I think, is less likely than the third option, which is that of chronic infection within humans in the context of suboptimal immunity, allowing the virus to persist, generate large amounts of diversity in different compartments, and then potentially those individuals then receive treatments such as monoclonal antibodies, convalescent sera, and even drugs, and then this allows the virus to adapt intra-host before spilling out. So this is the option that I favour, and I think most scientists do favour this possibility. And really, our foray into this area came very, very early on, and this study is really the reason we got into SARS-CoV-2. And it's because we were asked to discuss a patient in the hospital who had a chronic infection with SARS-CoV-2. Here you can see on the timeline up to 100 days. On the x-axis, you can see different treatments that the patient was given. He had been diagnosed with lymphoma 10 years previously, was currently using rituximab, which is a B-cell depleting agent, which renders your antibody levels very low. And this had allowed a persistent SARS-CoV-2 infection. What you can see here is the CT value of the infection. And along here you can see that the patient was given remdesivir twice and then received convalescent plasma at day 65, and then a third dose of remdesivir out here at day 93, as well as convalescent plasma. And what you can see here in the phylogenetic tree is how the virus within the individual had evolved over that time period. There's quite extensive diversification with acquisition of spike mutations here in this branch, in particular delta 6970 in the internal domain, and a change at 796H. Both of these mutations are seen in the Omicron BA1 variant. And of course, this was a case taken from 2020, well before Omicron had emerged. If you look at the deep sequencing data over time, this is just from days 82 to 101. You can see that there's an immense amount of change going on. If I had shown you the data from day 0 to day 60, there was virtually no change. These would have all been flat lines. But something happened after day 82. Essentially, this was following the infusion of neutralizing antibodies in the form of convalescent plasma. You can see selection of groups of mutations, and this is the variant frequency on the y-axis. So these are low-frequency variants are up here. They never reach fixation. They come and go. But there are some mutations and groups of mutations that reach fixation at nearly 100%. And this is indicative of haplotype competition. In other words, different strains of virus within the individual are competing against each other over time. And the important thing here is that the dominance of those is alternating. So the selective pressure that we were applying through the convalescent plasma, we believe, was actually applying a pressure, but then as the neutralizing antibodies disappeared, different populations of virus were regaining control of the infection. And so what you have here is evidence of real-time evolution. But you can see here the 796 and 6970 changes that are going up and down. And so we wanted to know, of course, at that time we didn't know anything about these mutations. So we wanted to know what they were doing. The 6970 is in an exposed loop in the N-terminal domain out here. The receptor binding domain is up here. And the other change is in the S2 fragment down here. Again in a surface loop that's exposed. And again, both of these, of course, eventually found their way into the Omicron variant. So we used our expertise in HIV virology to address what these mutations were doing. We made pseudotyped particles expressing the spike protein, and we made the mutations that we'd observed in the patient, both in singly and the combination. This is a Western blot showing you that those virus particles were HIV inside because they have P24, but also they're expressing spike, and of course they're well expressed. Looking at infectivity here, you can see that the 796 change leads to quite a big drop in virus infectivity. So this was quite surprising, but the other mutation led to a modest increase in infectivity. And we realized what was going on when we looked at how these mutations affected the virus's ability to escape antibodies. So what we did is we took the plasma that we treated the patient with that contained all of these mixtures of antibodies, and we mixed it with the virus and then examined how well those viruses were able to infect a target cell. And so if there were neutralizing antibodies present in the sample, they would block infection. And what we found is that the 796 change over there actually led to partial resistance to the neutralizing antibodies, and the deletion here had actually no effect. So we realized that we'd found two mutations, one of which was leading to immune escape, which is 796, and the other mutation was compensating for that loss of infectivity you can see here by increasing the infectiousness of the virus particles. And so that's, again, a paradigm for how this virus is working. Some of the mutations are there to escape our immune system, but some of those mutations lead to detrimental effects on infectivity, and the virus then needs to make other mutations in order to compensate for that. So with one patient, we'd actually sort of approached some key paradigms of the virus. Other work, of course, there are a number of other reports of chronic infections, but here's a really important one from colleagues in South Africa, where in an individual living with HIV, they documented an infection over 6 months, and these are the mutations that were accumulated over time, much, much more extensive than in our individual, and a large proportion of these mutations are present in the Omicron variant. So they observed this happening over time, and this is why we believe that variants of concern likely arise in chronically infected individuals, and HIV is potentially a big risk factor. This antigenic map here shows you that the virus that the patient was infected with initially had shifted to over here, becoming much more like a beta virus, largely because of mutations such as N501Y and E484. So you could see, again, this sort of antigenic shifting of the virus using serum neutralization assays that mirrored very nicely the mutations that were being accumulated. So that's an example of how why we believe that chronic infection is causing or is the source of new variants. The problem here, of course, is that the pool of immune-compromised individuals is enormous. It ranges from diabetes through to chemotherapy patients, through to cancers, of course. So this is a huge problem globally. HIV may be a significant risk group, but we do need more data for this. And of course, the way to limit new variants emerging is to control the total number of new infections. Okay, I just want to spend a couple of slides just talking about alternative routes to diversification. One is recombination, because a lot of people have been discussing this, and in HIV we know recombination is a major source of diversity in that virus. There were some early signs that recombination was happening. Here, a paper discussing recombination between alpha and its basal lineage. More recently, there were deltacron recombinants described. But none of these were very successful until XBB came along, and of course this we believe is the first very successful recombinant virus, and it's the result of recombination between two BA2 lineages that have had a swapping event essentially in the receptor binding domain around here, and you can see that the XBB variants, of course, are very, very resistant to neutralizing antibodies generated through vaccination and past infection. So XBB variants have recombined and generated viruses that are poorly sensitive to neutralizing antibodies. Another really interesting and important, we believe, important source of genetic diversity is potentially from the host. I said earlier that the virus has proofreading activity in its polymerase, and so again, the paradigm of viruses generating diversity is that they make mistakes when they copy their genetic material. That is certainly true for retroviruses, but it's harder to understand how that could be at play for something like SARS-CoV-2, which has a polymerase that does proofreading. And in fact, it may be antiviral molecules in the host that drive some of the diversification of the virus. And there's a very recent paper here from David Robertson's group that has looked at mutational signatures in SARS-CoV-2 sequences from around the world and looking at different nucleotide contexts. These are triplets. You can't see it here, but these are different combinations of bases. And you can see that that there are signatures of APOBEC mutation or ADAR-like mutation. So this is adenosine deaminase editing. So you can see here that there's a different signature for APOBEC and a different one for ADAR, but they're all seen in SARS-CoV-2 genomes from around the world. So this may be, the host may be driving some of the mutational pressure on the virus, and of course the selection pressure then enables those to be fixed. So we're learning more and more about diversification of this virus even now. Okay, so I'd like to spend a few slides now just talking about what's the result of all of this evolution, what are we actually observing. And I'm going to take a slightly historical perspective because I think it's important for us to understand where we've come from. And initially, of course, we had these viruses that had N501Y. We all remember alpha, beta, and gamma. They all emerged within a short time frame of each other or were identified in different parts of the world, South Africa, Brazil, and of course the UK. They all had this signature mutation, and we believed that this was how all variants were going to arise. Importantly, 484 and 417, again seen on these two viruses and again seen in Omicron. So these are key escape mechanisms for the virus. Very early on with alpha, we were able to show, using neutralization assays, that in fact the alpha variant of B117 was very similarly sensitive to neutralizing antibodies generated after people had been vaccinated, and that was very reassuring. These neutralization figures essentially show you how sensitive the virus is to neutralizing antibodies, and the higher the number, the more sensitive the virus is, and the lower the number, the more resistant the virus is. So you can see here that B117 is very similar to wild type, about a 1.5 to 2-fold loss, but not hugely significant. But other viruses, such as the beta variant and P1, which is the gamma variant, showed more elevated degrees of resistance to neutralizing antibodies. So that was early on. Delta variant turned that paradigm on its head a little bit because this variant did not have N501Y. It had a different set of mutations in the receptor binding domain, notably L452R and 478, and importantly a cleavage site mutation, which we'll discuss a little bit later, and then a group of mutations in the NTD that hadn't really been seen before at positions, in particular 156 to 158. And this virus here, termed at the time B617.2, showed a reduced neutralization sensitivity, in other words, more resistance to neutralizing antibodies in sera from vaccinees. And importantly, the Delta variant really was the first virus that demonstrated that breakthrough of vaccine could occur. And we were fortunate enough to team up with colleagues in India, where AstraZeneca had been scaled up and people had 2 doses. And this is the number of days since the second dose. And this is a phylogenetic tree showing you infection within a hospital and the different lineages that were represented there. This was a period of mixed lineage circulation, including alpha variant, the B lineages, and then the Delta variant. And what was really striking here is that the Delta variant here was responsible for these clusters of breakthrough infections. In other words, when we did this formally, we found that the Delta variant was much more likely to lead to a breakthrough of vaccine-induced immunity than the other variants. And this reflected the fact that it was more neutralization resistant. I believe also that this was partially due to the infectivity advantage of Delta over Alpha, which I'll show you in a moment. So really, we didn't really think that vaccines could be broken through until this happened, and it's important for us to I think, take ourselves back to those days. And in fact, following these data, the CDC immediately issued advice for people to mask after they'd been vaccinated, which I'm sure you'll remember. We then went on to show that these these Delta variant transmissions could be transmitted by fully vaccinated individuals using, in collaboration with Chris Illingworth, we were able to show through genomics and epidemiology that fully vaccinated individuals infected with Delta were capable of passing the virus on to other individuals. And again, that I think was an important public health contribution. Fast forwarding now to Omicron, we again was hugely surprised by the fact that when we looked at Omicron and we looked at neutralization, and if you compare the second dose neutralization in blue here, the 50% serum neutralization is around 1,000 for Delta, but only around 100, so it's tenfold more resistant to neutralizing antibodies generated by vaccination compared to Delta. That was a huge step, really. But actually what was potentially more surprising was that if you look at the booster dose, the third dose of vaccine, this rescued or enhanced your neutralization of Delta, but also to a very significant extent for Omicron, despite the fact that you were boosting with a Wuhan virus. You know, and the Omicron variant had around 50 added mutations, about 25 of them in spike. So boosting neutralizing antibody levels actually could compensate for the fact that you had a highly divergent virus. I've shown you some data for evasion of neutralizing antibodies, but now, of course, we're also becoming more aware of the fact that the virus is also escaping from innate immunity. Work from Greg Tau's lab here has shown that the alpha variant is upregulating transcripts in ORF9b, ORF6, and N, and protein levels in order to overcome innate immunity and interferon signaling. This has also now been seen in Omicron lineages, and there's a talk later on today that will address the adaptation of Omicron lineages to escape innate immunity. Just like to turn now to examining infectivity and replication. I think a lot of focus has been on immune evasion, and that's what you hear about in the news, really. But actually, I think that an equally important story for this virus is its ability to increase and optimize its replication and life cycle. So we were able to show early on when Delta emerged, this 617.2, that it was much, much more infectious than the alpha variant in a number of systems. These are Cali3 lung cells. You can see that the Delta variant here is in black, and work done in collaboration with Wendy Barkley shows here that in human nasal epithelial cultures mimicking the upper airway, you can see the Delta variant was significantly higher in titer than the alpha variant. So this virus had clearly adapted to become more infectious. You can also see this in airway organoids in green versus orange here. So globally increased replication in different cell types was a feature of the Delta variant. And in addition to that, you can see in terms of what the mechanisms are for the replication advantage, we know also that the binding affinity for, between spike and ACE2 has also been increasing over time. Here you can see bilayer interferometry data for Wuhan, Delta, and Omicron. And this is a protein-based assay that looks at how well two proteins are binding. And there's an on rate here and an off rate. And I hope you can see here that the off rate is something that's been increasing as you go through the lineages. So you can see that here you get dissociation between the proteins, but with Delta you can see that the proteins bind for a longer period, and Omicron they bind for even longer. So that prolonged contact between ACE2 and the spike protein gives the virus a better chance to get inside the cell. There are other more interesting ways that the virus is increasing its infectivity. For example, in nucleocapsid, we noticed early on that in different lineages, there were changes occurring in nucleocapsid at positions 202 to 204. And if you make those mutations in vitro, you can show that the number of genome copies goes up quite significantly, and viral infectivity goes up significantly. So the virus is also ramping up its replication, not only through spike-mediated entry, but also using other genes such as nucleocapsid. Turning to tropism of the virus, we had a really nice discussion about tropism yesterday for influenza, and Wendy also then proceeded to discuss tropism of SARS-CoV-2. We've worked quite extensively in this area, and I believe it's a really significant event for the evolution of SARS, because this may be one of the things that explains why the incidence of pneumonitis and severe hypoxia has been reduced with the advent of Omicron. So Omicron emerged at a really interesting time in that it preceded Delta, and really these two viruses have shown us the opposite ends of a spectrum, as it were, for tropism. So this figure just shows you the mutations in Delta here on the left and Omicron on the right. You can see Omicron has many, many more mutations spanning the NTD, RBD, but also the cleavage site between S1 and S2. And I'd like to spend a couple of minutes just talking about spike cleavage. This is a process that is required for full maturation and entry by the plasma membrane route of the virus. It happens in the producer cell. The spike is produced as a full-length polypeptide, and furins, which are host proteases, cleave the spike protein here at the S1, S2 junction. And this furin cleavage site has been changing during evolution of variants of concern. The importance is that once that cleavage site has been cleaved, it allows the virus to engage TMPRSS2 on the target cell. And once TMPRSS2 is engaged on the target cell, it is then able to enter. If TMPRSS2 is not engaged, the virus needs to be taken up by endocytosis, and that points to the essential role of the S1, S2 cleavage site. So in terms of cleavage efficiency, we've been very interested in how that's been changing over time. If you look at the alpha variant here, this Western blot shows you the spike protein. This is the full length up here, and this is the cleaved S2. You can see that with Delta, there's a much more intense band for S2, suggesting that Delta is cleaving its spike protein more efficiently, and this potentially is related to the fact that in the furin cleavage site here, the alpha has an H at position 681, and Delta has an R, again, positively charged residue. So why is that relevant? Well, because when Omicron came along, we looked at that cleavage region. We found 681H, but we also found two other mutations at 655 and 679. We predicted this would be a very highly cleaved spike. It would be very good at using the TMPRSS2 mediated pathway for plasma membrane entry. But in fact, we found that the cleavage of Omicron actually was diminished in relation to Delta, despite having multiple mutations in that region. And therefore, we postulated that the virus would not be able to enter via the plasma membrane by engaging TMPRSS2, but would instead be forced down this endocytic pathway where TMPRSS2 was not required, and then infect the cell through that pathway. As we discussed yesterday, the endosomal entry route is dangerous for a virus, because there it's going to encounter host defense proteins such as the IFA-TEMs. And so to test this, what we did is we took a range of cell lines and organoids expressing different levels of TMPRSS2. On top here are the TMPRSS2 high cell systems that we used, and if you infect cells with pseudotyped viruses, what you can see is that the Delta achieves very reasonable levels of infectivity, but the Omicron is 1 or 2 orders of magnitude diminished in those TMPRSS2 high cells, so showing you that the Omicron variant is not efficiently engaging TMPRSS2. If you look in cell models where there is low levels of TMPRSS2, for example, these HeLa ACE2s, 293s, or even H1299s, you can see that Delta and Omicron have nearly identical infectivity profiles. So the level of TMPRSS2 appears to dictate the entry efficiency in those cells. And if you plot the TMPRSS2 levels here, this is based on mRNA expression, you can see that there's a degradation, and the high infectivity, the Delta variant infects these ones very well, and the Omicron variant will infect these equally, but Omicron is impaired in these TMPRSS2 high cells. To test that further, we used drugs specific for those two pathways. So Delta is in orange and Omicron is in gray. If you use Camostat, which is a TMPRSS2 inhibitor, you can see that Delta is almost completely inhibited to 100% as you titrate that drug in, suggesting that Delta requires TMPRSS2 for optimal infectivity. But but Omicron is not completely inhibited with that drug. If you try to block the endocytic pathway using E64D, that's a drug that prevents acidification of the endosome. You can completely inhibit, you can quite effectively inhibit Omicron, but you can't inhibit Delta very well. So the sort of summary of those data are that the cell entry pathway preference has been altered. Omicron is entering via the endosomal route, and Delta is entering via the surface membrane route. And I showed you earlier that TMPRSS2 is enriched in lung organoids and Calu-3 cells, which are representative of the lower airway. And we believe that the TMPRSS2 low cells are more representative of the upper airway. So this is adaptation to a different cell type and a different part of the airway that potentially explains why Omicron has become less severe in terms of generating hypoxia. Of course, it's still generating a lot of inflammation, which triggers thrombotic events, strokes, heart attacks. So it doesn't mean that the virus is mild overall, but it just does mean that this explains why we're seeing lower rates of hypoxia and lung involvement with Omicron. I want to spend a couple of moments talking about allostery because I think this is a critical topic in that we are trying now to use genomics to understand the phenotype of the next variant. We're constantly trying to do that, and we showed you with Omicron that we were wrong. We saw all those mutations in the cleavage site. We expected it to be very well cleaved and use TMPRSS2 very well, but it was the opposite. So we think that allosteric interactions, which are long-range interactions between different parts of a protein, are important. And we did some experiments to swap the N-terminal domain, which, if you remember, is sitting on the side of the spike protein. We tried to swap that between different variants to see if we could transpose certain qualities onto different viruses. And the reason we did this is because we found that the N-terminal domain from the Delta variant appeared to be very good at conferring entry in these lower airway cells and the airway organoids. In other words, the NTD from the Delta variant may potentially increase the cleavage efficiency at the S1-S2 junction. And if you remember from the figure earlier, the S1-S2 junction is a long way from the N-terminal domain. And so we tested this, and you can see, of course, yes, that if you take the Kappa variant, which was a relative of Delta at the time, and you stick on the Delta variant NTD, you can increase its usage or its infectivity in these TMPRSS2 high cells. And if you remove it from Delta and replace it with a Kappa N-terminal domain, you end up with a diminished use of TMPRSS2. But in cell lines that have low levels of TMPRSS2, you can see that none of those domain swaps have any difference whatsoever. So this was interesting and intriguing. We then proceeded to make single mutations of that N-terminal domain that were found in Delta, and you can see that actually each one of those mutations has a significant effect on infectivity in the TMPRSS2 high cells, but very little effect in cells that don't express high levels of TMPRSS2. So we're showing you here that mutations within the N-terminal domain, again sitting quite a long way from the cleavage site, are able to impact structure such that it will change the tropism for certain cells. We then looked at the cleavage event itself, and you can see here these are the Western blots of these transposed viruses, and if you quantify those, you can see that that swapping the Delta N-terminal domain onto Kappa does increase the cleavage efficiency overall. So we ended up with an intriguing sort of observation that allosteric interactions can also determine the tropism of a virus. So if we're going to try and predict whether the next variant is going to be a lung tropic virus or an upper airway tropic virus, we're going to have to become much more sophisticated in our approach. And to add to the complexity, if we try to make Omicron more TMPRSS2 sensitive, in other words, we try to make Omicron like Delta, again using pseudotype viruses, so these are not gain-of-function experiments, but we transposed that Delta N-terminal domain onto BA1 Omicron, and we were not able to increase the infection in TMPRSS2 high cells. And really that was not surprising because, of course, allostery is sequence-specific, protein-specific. So putting the Delta NTD onto a very, very unrelated virus was not necessarily going to yield the same result as transposing it onto kappa, which was of the same lineage. So this tells us that although allosteria is important, but local sequence context is also critical. For the last few minutes, I'd like to really talk about syncytia formation and cell-cell fusion, which arguably may be one of the most important aspects of SARS-CoV-2 evolution and pathogenesis, although it's become quite difficult to study. Now, syncytia formation is something that many enveloped viruses do. So the glycoprotein from viruses is able to cause fusion events between neighboring cells. So just the spike protein expressed will enable two cells to fuse together. And this is known for a number of viruses, such as HIV and RSV. These are data from HIV-infected macrophages. So you can see these syncytia forming here. We believe that this is a function of the spike protein that's potentially been favored through evolution because it allows the virus to spread from cell to cell without encountering the extracellular environment, in other words, able to then escape antibodies, for example. And our colleague Leo James developed a split GFP system for studying this in vitro for SARS-CoV-2. Essentially, you split GFP into two and you mix two different cell populations, and if the spike protein expressed in one cell is able to bind the ACE2 in the adjacent cell, they will fuse, and then the GFP will be expressed, and the cells will go green. So this is quite an elegant system for testing whether spike proteins that are introduced into a cell are able to mediate cell-cell fusion. Why is cell-to-cell fusion important? I mean, I've mentioned that it might be important for the virus, but there are a number of studies that are implicating cell-cell fusion and syncytia formation in pathogenesis. So a number of autopsy studies have shown that in lungs of people who have died, there are large syncytia in the lung tissue. Studies such as this suggest that syncytia may engulf lymphocytes, for example, and lymphopenia, you may recall, is a cardinal sort of sign or finding in SARS-CoV-2 infection. Other studies suggest that cell syncytia formation may trigger pyroptosis, for example. So we believe that this is relevant for SARS-CoV-2 pathogenesis. And we were really struck by this finding, which is that Omicron is highly impaired in the ability to induce cell-cell fusion. If you look at the Wuhan 1 virus here, you can see that there's a lot of green, which means that lots of cells have fused together once the spike protein from Wuhan 1 was expressed in those cells. But if you express the Omicron spike in those same cells, you get very, very few fusion events. And if you quantify this over time, you can see here that Omicron leads to very, very low levels of cell-cell fusion in comparison to wild type here in black, or Delta, which is is significantly enhanced over wild type. So there's a huge difference here between Delta and Omicron, which may also feed into the difference in pathogenesis that we observe clinically. So it's been very, very difficult to tease apart those two functions because we believe that cell-cell fusion is also TMPRSS2 dependent. The N-terminal domain I mentioned earlier could confer use of TMPRSS2 in certain contexts. Intriguingly, we and others are finding that the N-terminal domain can also affect syncytia formation. A paper here shows that if you use 4A8 antibody, this is an N-terminal domain targeting antibody, so if you titrate that in, you can block fusion of cells for SARS-2, but not SARS-1. Of course, SARS-1 does not generate syncytia, but SARS-CoV-2 does, and this can be blocked through antibodies against the NTD. We've also shown that in the alpha variant here, you can abolish cell-cell fusion by repairing that delta 6970 deletion that I told you about earlier. So this is an N-terminal domain deletion, and you can see that it does affect cell-cell fusion. So the story is much more complex than simply ACE2 binding the receptor binding domain and leading to cell-cell fusion. Different domains of the spike protein. I've shown you the N-terminal domain has a role here. I haven't had time to show you that the cleavage sites will also have an effect on that efficiency. So I'd just like to summarize this part of the talk by really emphasizing that the Omicron emergence is a radical event. It's so significant that I think that we haven't fully grasped it yet. Not only were there multiple new mutations and huge amount of immune escape, but this tropism shift really has changed the character of the disease. You can see here, of course, this is the hospitalization rate for Delta and Omicron. This is early on and data from South Africa. These are mouse data showing you again that Delta is less pathogenic in animal models. In airway organoids, you can see here there's a huge difference in infection of those organoids that represent the lower airways. But in nasal epithelia, you can see very, very similar replication kinetics as Wendy showed you yesterday. So this is a major event. And really, I think that it's really important for us to spend time figuring out how this all happened. What drove that biological change that gave us a virus that appears attenuated and that fulfills, the sort of paradigm that viruses become less pathogenic over time. I think that this is an evolutionary phenomenon. I don't necessarily believe that this supports the fact that viruses evolve to become less pathogenic. But knowing about the negative consequences of immune selection and escape on the virus could inform effective therapies and vaccination approaches. In other words, can you force viruses into these dead ends or these evolutionary pathways that render them less pathogenic? And of course, this is borne out by the fact that if you've got all these different VOCs, but now all the VOCs that we see, or all the variants, are relatives of Omicron, telling us that the virus has found a way of becoming massively infectious and also able to generate large amounts of diversity in order to generate lots of different offshoots, including recombinants now. So how does this feed into what we do about it? Well, vaccination has a number of challenges. We did some early work here on first dose Pfizer vaccines, and we showed that if you were over 80, there was quite a big drop in your neutralization of virus, and that was largely related, we think, to reduced degrees of somatic hypermutation and B-cell maturation, as well as T-cell help. This persists into dose 2. You can see here that, if you have more than 80, you've got a reduced neutralization across the board for different variants, beta, gamma, and alpha. We believe that after the third dose, things do start equalizing, and of course you've got natural immunity on top of that. But of course, we've also had to deal with the waning efficacy or effectiveness of vaccines over time. These are data from when Delta emerged following the Alpha variant. You can see that the effectiveness of against all variants falls over time. This is common knowledge now. So we need to find ways of overcoming that. Boosting, of course, has some effect, but of course you get waning again. So these are data from dose 4 homologous vaccination. We've started using bivalent boosters. Of course, these were touted as the next thing, but of course, the neutralization improvement here at 3,000 is only 1.5 times what you would have found if you used just the Wuhan vaccine. So we need new ways of vaccinating. Do we target S2? This is a nice paper showing you that S2-based vaccination may be a way forward because it's highly conserved amongst coronaviruses, and you get reasonable neutralization when you immunize against that region. There is still work going on about RBD-based vaccines. You can find highly conserved regions of the RBD and then generate immunogens based on that, and it is quite successful at neutralizing different viruses. So there are a number of different approaches being used, but we don't know where we're going to be in a year's time. So just to summarize, variants likely arise from chronic infections. Variants have compromised vaccines, as we know, and Omicron has shifted its biology, and we don't know if this is the end or whether there will be a new variant. We can get high protection from severe infection following 3 or 4 of homologous vaccine doses, and that's really reassuring. But for the future, we really need to be looking at how to extend the interval between doses. We need to know how to deal with new variants. Is it through multivalent vaccines or targeting other parts of the spike protein to develop pan-coronavirus strategies? And then, of course, there's the big story about nasal immunity and nasally delivered vaccines. But the problems here, of course, are going to be about durability of protection. So with that, I'd like to thank my team and the collaborators on this slide and many others who may have not been mentioned, and thank you for your attention.